let's discuss with the next type of reversible inhibition, that is non-competitive inhibition or non-competitive inhibitors. Now, what do you mean by non-competitive inhibitors? This can be very well explained if you see this diagram carefully. Now, here you can see this is the enzyme. Okay, can you see this is the enzyme and this is the active site of the enzyme. This. Okay, not this is the active site of the enzyme. This V shaped is the active site of the enzyme. Now this is the substrate molecule which also has this triangular part and it can easily fix into the active site of the enzyme and it can form enzyme substrate complex. And here there is an inhibitor. Now you can see substrate and inhibitor, they are not having any similarity in their structure. So there is no competition between them for the active site. But what happened here, this inhibitor, it attack on the enzyme from other side. It is not the active site, but it attack from the allosteric side. Allosteric means other side. And when this inhibitor attack the enzyme from other side, it brings about some structural changes in the enzyme structure in the active site of the enzyme. Now, as soon as this inhibitor attached to the enzyme, can you see the active site, its shape and structure get changed. Now, substrate cannot attach to the enzyme, and as there is no enzyme substrate complex formation, no reaction takes place. Here inhibitor, it doesn't bind to the enzyme from active site, but inhibitor bind to the allosteric site or other site and it changes the shape of the active site. So, as per name, now here there is no competition between the substrate and inhibitor, which we have seen in competitive inhibition, because here inhibitor bind to the site different from the active site, and when it bind to the other site, it causes the change in the overall three-dimensional shape of the enzyme. Now the enzyme, it may bind to the inhibitor, the substrate, or it can bind both together, but it doesn't lead into the product formation. Now, the effect of non-competitive inhibitor cannot be overcome by increasing the substrate concentration. In competitive inhibition, if you remember that as there is a competition between substrate and inhibitor, because both looks like same, if the concentration of substrate is increased, then that competition can get overcome and substrate can easily attach to the enzyme and rate of reaction is increased. But here, in case of non-competitive inhibitors, as substrate and I will just clean this, substrate and inhibitor, they are not structurally alike, so there is no competition between them. So there is no, but what happened when inhibitor attack on the allosteric site of the enzyme, it changed the complete shape of the active site. So if the substrate concentration is increased here, it has no effect. So effect of non-competitive inhibitor cannot be overcome by increase the substrate concentration, and hence there is a decrease in Vmax. Now see here, what is the effect of non-competitive inhibitor on the Vmax and the Km value? That can be very well explained if we see the plot, Michaelis-Menten plot, and as well as Lineweaver-Burke plot. Now here in this plot, when there is a plot between substrate concentration and velocity of reaction, here this hyperbola is for only substrate or when no inhibitor is present. This is the Vmax. This blue line, it indicates no inhibitor is present, and this is the Vmax, and this is the half of the Vmax if no inhibitor is present, and when there is velocity is half, then that is the value of Km that we have already seen. Now see the red plot. This red plot, it is for the reaction in the presence of reversible non-competitive inhibitor. Now when inhibitor is present, the rate of reaction is decreased. At the same time, Vmax is also decreased. See, a red color line. So this is the Vmax for the inhibitor, and as this is the Vmax, now this is the half of the Vmax, and we know half of the Vmax, it is called as Km value. So the substrate concentration at half of the Vmax is Km. Now from this complete plot, you can understand that in non-competitive inhibition, Vmax is decreased. There is a change in the value of Vmax, but Km value remains same in both the cases. Here, this is a Lineweaver-Burke plot when we plot a graph between reciprocal of substrate concentration with the reciprocal of the velocity. Then we get a Lineweaver-Burke plot. Here also you can see that Vmax is changed, but the Km value, it remains same. Some example of non-competitive inhibitors are heavy metals like Ag+, Pb2+, Ag2+ can non-competitively inhibit the enzyme by binding with cysteine sulfahydryl groups. Cysteine, it is a sulfur containing enzyme, amino acid, and if these heavy metals, if they combine with the sulfahydryl group of cysteine, then they can inhibit the activity of the enzyme, but it is the non-competitive inhibition. Glucose 6-phosphate inhibit hexokinase enzyme in the brain, and this inhibition is also non-competitive inhibition. Now let's discuss the third type of reversible inhibition, that is uncompetitive inhibition or uncompetitive inhibitors. First we have seen competitive, second is non-competitive, and third one is uncompetitive inhibition. Now this uncompetitive inhibition we can very well understood by seeing this reaction. Now in this reaction you can see that here that there is a enzyme plus substrate, there is formation of enzyme substrate complex. Now here you can see inhibitor is not attacking on the enzyme directly like in competitive and non-competitive. Here inhibitor is attacking on the enzyme substrate complex, not directly on the enzyme, and when it attack on the enzyme substrate complex, there is a formation of new complex, that is enzyme substrate inhibitor complex, and there is no product formation. So, if this reaction is normal, then what? Enzyme plus substrate gives enzyme substrate complex, which result into enzyme and product. We know these reactions for enzyme catalyst. But when there is an uncompetitive inhibitor, it doesn't compete with the substrate by any means. It is not attacking on active site or on allosteric site. But what it is doing? This inhibitor, it is attacking to the enzyme substrate complex. And once it attack on the enzyme substrate complex, as I told you, new complex is formed which doesn't go in the product formation. So we can define uncompetitive inhibition, which is also known as anti-competitive inhibition, takes place when an enzyme inhibitor or inhibitor binds only to the complex formed between the enzyme substrate or ES complex. Uncompetitive inhibition typically occur in reaction with two or more substrate or products. If two or more substrates are there or if two or more products are there, then such type of uncompetitive inhibition is observed. One thing you need to remember that uncompetitive inhibitor is not attacking to the enzyme directly, but it is forming complex with the enzyme substrate complex. Now, these are the plot and Lineweaver-Burke plots for uncompetitive inhibition. Now, in these you can see here, this is red line is for inhibitor and blue line is the no inhibitor. In both the cases, Vmax is changed. This is the Vmax maximum value for inhibitor and this is the maximum value for this. In both the cases, maximum velocity is changed, Vmax is changed, and as Vmax is changed, Km value, this is the Km value for the substrate and this is the Km value for the inhibitor. So both the values are changed. So we can say that there is a decrease in the Km value for the substrate and there is again decrease in the Vmax value. The same we can see here. Red line is for inhibitor and blue line is for no inhibitor. In uncompetitive inhibition, Km is reduced as well as Vmax is reduced. Both the values are reduced. Here, all the three reversible inhibitions and their plots are shown here combiningly. So, reversible inhibition, it is of three types: competitive, non-competitive and uncompetitive. In non-competitive, now these are the direct plots and these are the double reciprocal plots, or we can say these are the Lineweaver-Burke plots. We have already discussed all these plots in detail, but still here you can see when in case of competitive inhibition, when there is a competition between inhibitor and substrate to attack on the active site, in that case Vmax is unchanged but Km is increased. Km value is increased. Now, when there is a non-competitive inhibition, in that case Vmax is decreased. When inhibitor is there, Vmax is decreased but Km remains unchanged, while in uncompetitive inhibition, both Vmax as well as Km, both are decreased. The same we can explain by using these Lineweaver-Burke plots, which we get when we plot a reciprocal of concentration of substrate to the reciprocal of velocity. Here you can see Vmax is constant, but Km is change, Km is increased. Here Km is constant, but Vmax is decreased or Vmax is change. Here both Km and Vmax both are decreased. So here we have finished about one type of inhibition, that is the reversible inhibition. And reversible inhibition, it is further divided into three types, that is competitive, non-competitive and uncompetitive inhibition. Thank you so much.